So welcome everyone to the Brain Space Initiative kickoff. I just wanted to kind of remind everyone that we'll be in kind of listen-only mode for the initial talk, and then we'll do some questions and answers at the end. Any questions you can always submit via chat. And also as a reminder, this will be recorded and shared later. So. Let me go ahead and share my screen. Okay, can you see that, Armin?
Yes, very good.
Sounds good. So I'll go ahead and get started then. Let me minimize this. Okay, so the Brain Space Initiative, what I'm calling it is a social and technical resource for advancing brain research. It's basically the brainchild of Armin, who has been talking to me for some time about doing something like this. And so you can blame him, but I think this is a really cool initiative, and I think that there's really a need for it. And let me try to outline why I think that is. First of all, as we all have kind of noticed, especially in this time when we're all virtual, there's just tons of information out there, and really trying to figure out how to read within the waves of that ocean of information is really challenging. People tend to get caught into sort of set ways of doing things. There are lots of silos, and we want to try to break those down. I really think there's a lot more need for more plurality in analysis approaches, comparisons among approaches, development of new approaches. There's also a gap, obviously, as we know, between more junior and more senior folks, and this is really meant to be kind of a non-threatening way to have discussions and engage. And very focused on diversity and inclusiveness, regardless of race, background, nationality, et cetera. We want this to be fully open. And we want to encourage interactions, presentations of results, discussions, no strings attached, and provide the opportunity to form groups. So why do we need it? Well, we have lots of virtual talks we can go to, but those are typically, you go and then you're done. There's not a lot of follow-up or interaction most of the time. Hackathons is another really nice approach. Those are intense. They can lead to follow-up projects, but they also tend to fade away after that intense working period, with the exception of maybe one or two groups. Courses are very useful for learning the background, hands-on, but you don't have this sort of ongoing social resource when you especially after you follow up in the course. It can be hard to break into a discussion group or find a place where you want to share results, just get feedback, ask silly questions, etc. And as we know, team science is becoming more common, but it can be still a little bit hard to interact, especially because you often have for team science, some very strong folks leading a group, and then there's like thousands of people involved, and it can be hard to kind of have a voice in that context. And so we're hoping this will be a way to sort of bridge some of those gaps. So there are lots of things that are planned. We're not going to do everything. We're just getting started here. We don't want to do too much initially. Obviously, virtual talks. Okay, yeah, another set of virtual talks, but just hang in there. We're going to try to do things like share relevant papers prior to the talks, do more engagement of the speakers throughout. We're offering Slack study groups, which are meant to be community-driven, opportunity to explore different topics, share findings, share preliminary analyses. Don't just wait until you're completed with the project. And then journal clubs provides a way to sort of go deeper into just seeing what's out there, following some of the trends. And we also sort of want to see interaction between study groups. For example, if you're focused on a particular brain disorder and then you're focused on a particular analytic technique, and sort of getting those two sets of people together. And really multi-group projects, doing team science if if that is of interest, focusing on things like verification, comparison, aggregate studies, etc. The talks will be hosted at that link below. We've already got some great talks lined up from some luminaries in the field, as you can see right here. More details will be added and more talks will be added as we go along. The leadership team for the initial Brain Space Initiative is Armin, Jean, Lou, and myself. As I mentioned, sort of Armin is the one who really initiated all of this. And he and I have talked a lot about how we really want this to become something that grows into a community resource. It's not meant to be focused on just what we do here at Trends. It's meant to be broader than that. And so we want to see more people come in. We want to see leadership transition, things like that. Our advisory committee consists of Peter Bannattini, Simon Eichhoff, Tanya White, Helen Zwo, Conrad Cording, and Jessica Turner. Some very experienced and again, luminaries in the field that can provide guidance and insight. Some of the initial study groups are here and shown in white. And what I'm going to do next is just show some examples of some of these techniques for what based on what we've done here at Trends, just to kind of provide some examples, plant some seeds, etc. Not meant to limit us in any way. Each of these study groups is there needs to be a leader or one or more leaders assigned to that group that'll kind of sort of steer the conversation, they may kind of build journal clubs around these topics. We're looking at things like multivariate methods, dynamic connectivity, data fusion, imaging genomics, population neuroscience, deep learning, cognitive neuroscience, neurodegenerative diseases. I've got some other ones here shown in orange that might be good topics to add, or maybe there'll be subtopics within the existing groups. There are lots of other things that could be added here. And again, we want this to be something that people take as their own and drive forward. Let me start. I'm going to go through each of these initial study groups and give some examples. For each of them, there's some core questions that I've just listed on the initial slides here. Again, not meant to limit, just meant to start the conversation. This one is a little bit more methods focused. How can we leverage information from multiple data sets? And a data set could be multiple subjects, it could be multiple time points, it could be multiple modalities, etc. How can we implement them efficiently? Subspaces, for those of you that aren't familiar, you could think of a subspace as something like a set of brain networks or domains, or maybe links between a subset of domains between modalities. So how do we kind of move forward beyond just throw everything into a big model and see what comes out? So this one will be, I think, a really interesting one. It's obviously, I do a lot of blind source separation work. Rogers is going to be a great leader for this. He's very approachable and very good at providing, insights and introductions to complicated topics. So for those of you that aren't familiar, there is a lot of blind source separation being done in neuroimaging now. And essentially the problem, as stated, is we have a set of sources, conversations in a room. In this cocktail party problem, we have microphones recording those conversations. And we want to take the recordings and estimate what the original sources were. And depending on your assumptions, you get different models. So if we assume that it's a linear thing, meaning the further away the microphone, the softer it is, and we assume the sources are independent of one another, or the conversations are independent of one another, then we have an independent component analysis problem. So this is applied, I'm sure you know, a lot to study resting brain data. Here's sort of just an intuitive cartoon. Again, you may already be super familiar with this, but we have a movie, say a pumpkins on a step with candles, and we take that movie and we can do a spatial ICA decomposition of it, and it gives us these sort of maximally spatially independent sources and their temporal signatures, and we can see really quickly that candle 3 here went halfway through the night. So this has obviously been applied a lot for resting fMRI and task fMRI. And an example, one of the strengths of these kinds of approaches is they're fully data driven. So rather than assuming that we've got a region of interest that has coherent voxels in it, which is not true, that has to be enforced by the algorithm somehow. We have these components, and they are by definition coherent. They have a temporal signature that represents them. So each one of these I'll call a connectivity network, sort of, and then we can take those temporal signatures and look at their cross correlations, and that's called functional network connectivity. So it's basically functional connectivity using ICA time courses. One of the strengths of this is that we're looking at, this will come up in dynamic connectivity. If we know the sources are coherent, that actually gives us, that won't mislead us in the case where, maybe we plop down an ROI and we've got a mixture of two things within the voxels of that ROI. The FNC matrix gives you kind of a connectivity pattern like we see all the time, where you have strong correlations. Here we have it within visual networks, for example, default mode network, and we have weak anti-correlations with default mode. One of the challenges of ICA is that it is data driven, and so that you get sources out and you don't know what you're going to get necessarily. These networks are pretty replicable, but we get artifacts, we get sometimes other networks, etc. And so that process of figuring out what's good, what's bad, how do you label these, how do you group them and order them properly to get this structure in the matrix can require some expertise. There's some subjectivity there. Another alternative approach is to use something that is more sort of automated. And so we've been doing a lot of this recently. This one we're calling NeuroMark, which is essentially you take some templates, some component templates derived from large data sets. So here we have human connectome data and Gaussian superstruct data, 800 and 1,000 subjects respectively. We ran ICA separately and found replicated components and then used those to develop templates. So those are kind of like our atlas, but then the templates are used as priors in a spatially constrained ICA. So each individual subject gets a new ICA that is optimized and adapted to that subject. So you get subject-specific spatial maps and time courses that are already ordered and labeled based on these templates. And just to show you how well this works, here you have two separate data sets, and you can see sort of the main overall connectivity patterns are highly similar to one another. The things that we're mostly interested in are these subtle changes. For example, control patient differences. Again, these data sets both had schizophrenia patients and healthy controls within them, and you can see those patterns are highly similar for these two independently analyzed data sets. And we also compared, if we look at sort of classification, looking at region of interest versus ICA components, and we're finding, at least in this comparison, that the ICA approach is outperforming approaches that used, I think, AAL, and we used a Brainatome atlas here, and there's obviously lots of these now. So it's a very promising approach. Here, one of the benefits of it is it's completely subject by subject, so you can do cross-validation more easily, and you can do individual subject kinds of things. So this is an example where we're looking at a clinical application to predict medication response in youth that are either prescribed antidepressants or mood stabilizers. If we use that approach, we analyzed about 100 subjects. We did a cross validation, and we got, I think it was 95% accuracy in predicting medication class response based on folks who were followed up over the course of about 2 years using their initial brain scan. And that's a very relevant clinical problem, because often individuals do get put on the wrong medications and they can get ill as a result. Lastly, for this topic, Rogers will bring this up at some point, I'm sure, but we've been talking a lot about trying to bring together some of the existing approaches. There are lots of them. There's obviously independent component analysis, but something called independent vector analysis. There's subspace analyses, canonical correlation analysis, etc. There's a whole bunch of these, a bunch of acronyms, and it can be really confusing. Rogers has done a lot of work in trying to come up with a unified framework for modeling these kinds of things based on what kind of data you're looking at, whether it's one or more dimensions, whether it's one or more data sets. Dynamic connectivity. So there's been a lot of work recently, I guess it's over a decade now, looking at time varying connectivity. How can we estimate it? Strengths, limitations, what do they mean? How do we model spatial and temporal variations? This is a paper that we did recently. It was led by Dan Lurie, Dan Kessler, and myself, and really this whole group that met a few years ago at an OHBM Open Science meeting, and really got together and started sort of fleshing out what do these fluctuations mean, and what do we know now, and what don't we know, what are the limitations. And so I think this is a great resource to start to learn about this. Again, super basic stuff here. What we're trying to do is, instead of correlating, looking at functional connectivity, for example, between two time courses, which is really just cross correlation between them, we want to capture the dynamics, right? So maybe we have a strong correlation initially and a weak one later, and we can capture this by clustering into states. We basically estimate with a sliding window movies that vary over time, connectivity movies, cluster those into states, and then determine which subject is close to which state at different points in time. Again, there's a whole bunch of variations on this theme, lots of discussions about limitations and strengths and etc. One thing that Armin and I have been talking a lot about, as well as Robin Miller, are how to model spatial dynamics. So instead of assuming that your node is fixed, maybe it moves, maybe it grows or shrinks. And so those are some of the things that are overlooked, I would say, in the field. Not at least it's been a little bit unclear whether an approach is temporally dynamic, spatially dynamic, or both. And so in this paper here, we've tried to kind of categorize those a little bit. This is some work from Ashkan Fakiri, who is really trying to look at modeling more completely frequency. So if you do a sliding window approach, you're picking essentially a type of filtering to do on your data. And so he came up with this idea where you basically create a filter bank. So you model all possible, you model windows at any possible frequency. One example is you just sort of spread your frequencies across the whole spectrum equally. And in that kind of an approach, you get things, you can show that if you have slowly varying things that match the window, sliding window with a fixed window works great. But if you have higher frequency information, it gets missed, right? Because you filtered it out. But if you use this filter bank, you would still capture it. And so we can see, for example, connectivity states that are low frequency or broadband or high frequency, and we can look at comparisons with patients and controls, et cetera. Another way to look at this, sort of a different way to look at this, is to think about some work that's led by Hans van der Hoorn and Victor Vergara. This is sort of a dynamic attractor model. So this is not a dynamical systems, a full dynamical systems approach. This is coming at it from the data-driven side, but we're modeling not only the connectivity changes, but their derivatives. And so then we get these pairs that have, for example, similar amplitudes, but opposite patterned derivatives. So there's flow in and there's flow out. And so using that, you can develop these attractor models, and you can show that they map pretty well, at least into this particular data set where we were tracking recovery from traumatic brain injury. And we found that these attractors sort of disrupted early on, partially recovered, and then fully recovered. Last thing I'll mention in dynamic space is, if we didn't have enough already to look at, here you can search through connectivity values, you can search through windows, you can search through subjects, you can try to look at, for example, finding things that repeat, right, or that are similar across all of those dimensions. Challenging problem. This is work from Muna Raman, who was basically doing something called tri-clustering to find sets of connectivity patterns and subjects and windows. And if you do this and you do comparisons with group, you actually get increased sensitivity because you're comparing apples to apples. You're reducing the variance within these homogeneous subsets. This is some work from Apo Hyverinen's group looking at deep learning to essentially identify temporal primitives that are repeated in some of the human connectome data. And lastly, I'll just mention that we have a paper out that's meant to be sort of a survey of existing approaches, not just approaches from our group, but also introduces some of the tools that we've created in the GIFT software for doing dynamic functional network connectivity. Data fusion, okay, that's a very big space, but Zhenning Fu will be leading this. How can we combine multiple modalities? What are the best ways to link the data? Do we link it by looking at covariation, shared time space, latent space, et cetera? How can we validate these approaches? How do they compare to unimodal? You know, is there value added? One of the ways I like to think about data fusion is, you know, you can look at sort of something really visual inspection, two things side by side. Data integration would be like PET-CT fusion, where they just really co-register. Asymmetric fusion would be something like using fMRI to seed EEG sources. And symmetric fusion is something where you're looking maybe at covariation or, you know, sort of jointly estimating from the data. The obvious reason why we do this is because we believe that, you know, looking at, for example, separating two groups, it's possible that any particular modality might not have enough information to separate the groups. But if we look at this as a two-dimensional space and we look at it in this direction here, we can start to see things separate better. Okay. In terms of univariate versus multivariate, I do a lot of multivariate work, and primarily because we want to look for patterns. So instead of treating each point separately, we want to find, you know, sets of patterns that are sort of linked potentially between two data sets or two modalities. This is a figure, was an approach that was created by Sergei Pliss, essentially thinking about, you know, a carrot and a stick. So the carrot is, okay, do multimodal imaging, do multimodal analysis, because you do EEG and fMRI, you're going to find these really nice networks that match between the two modalities. You know, doesn't always happen, but it can happen. The stick would be, well, okay, if I do fMRI and MEG on this is an auditory oddball task, and I look at these two side by side, I can see that they're actually, you know, more different than the same, right? So what are we learning, right, from these two very different techniques? You know, if you have people that just do MEG, people that just do fMRI, they're going to, you know, make very different conclusions based on their data, and we need to spend the time to really try to integrate across. There is a global solution here, I believe, and so, you know, we're all studying the same thing. It's the brain, right? And so we're looking at it through different lenses, and we need to bring those things together. If you review the papers out there, you see that there's an increasing number of multimodal approaches. This is sort of a broader review, but just to kind of highlight that, you know, if you look at one thing, which is prediction accuracy, you generally find higher values in multimodal versus unimodal. We'll have to see whether that's, you know, that replicates, you know, issues of bias aside, but I believe it's true based on what I've seen in the data, and I think that that these papers are reflecting that. This is a really old approach now, but it kind of illustrates the point. So if we simply just jointly look at two different features, for example, an fMRI task map and an event-related potential collected with the same task but separate data sets, right? And then we look for covariation. We find maximally spatially independent, jointly spatially independent components that covary in a similar way across individuals. You know, this is an example from an auditory oddball task, and we get joint components out here that had an fMRI part and an ERP part. And once we do that, once we decompose that, we can remove the noisy components, keep the clean ones. They separate nicely here, into well-known ERP components. Here's N1, N2, P3 example, for example. And then we can, for example, take one particular point in time. We have the weights now, and we've got the modalities linked, and we can go back and reconstruct a weighted fMRI map for that point in time with using the ERP portion of the components. So we're sort of you're capturing high temporal resolution information and reconstructing the fMRI using these linked components. And it made a whole lot of sense. For example, if it's a little hard to see here, but we got initial auditory response, motor planning, then motor execution, and then this big P3 response here. So it made a lot of sense based on what we found here. And we can really leverage these modalities in a better way. There are tons of ways to do this. We're to blame for some of that. Here's an approach, though, from Jing Sui, which is looking at, again, it's looking at covariation by blending together the strengths of two approaches. Canonical correlation analysis, which is good at finding correlated sources, but if too many sources are correlated and it doesn't drop off cleanly, then accuracy suffers. Joint ICA is very good at capturing maximally independent components that covary. But if you add multiple data sets, it can start to degrade in terms of its performance as well. If we combine the two, we can actually leverage the strengths of both approaches and minimize the weaknesses. And so that's great. But again, it's fully data driven, so you get what you get when it comes out. We can go a little bit further than that and add an additional constraint, for example, with a reference, for example, cognition. And so in that sense, you're asking a question, identify within this high dimensional data linked multimodal components that are associated with a reference variable like cognition. And so one example of how we can use that is we took functional diffusion and structure data. We used a constraint to identify linked components. We showed that they replicated in different data sets. And then we went to new data after fixing these identified regions, and we showed that we can still find replicated prediction of cognition in unseen data. So again, I think it's a promising approach, and there are lots of variations on this theme and lots of different approaches out there. Imaging genomics is kind of another type of data fusion, but it's got it's really very different data, right? Because you really have a whole bunch going on in the genomics field that's somewhat distinct from what's going on in the imaging field. Some of the questions, so Jiayu is going to be a great leader of that group, and some of the questions are looking at cell-specific genetic signatures of brain abnormalities, temporal dynamics of genetic risk factors. Can we use this for intervention somehow? How can we leverage deep learning to do imaging genomic methods? There's initial discussions in the Slack channel about the first journal club as well. So again, motivation as before, joint modeling can improve our sensitivity. So we're trying to sort of combine across two modalities, but sensitivity is just a piece of the puzzle here. Beyond this, we get information about linking imaging features to genomic features. One approach we've used a lot is called parallel independent component analysis, and essentially what we're doing there is two ICA decompositions, one on, for example, a single nucleotide polymorphism array that's flattened into a vector, and a spatial map from structural MRI data, for example, gray matter. And we do those two ICAs, but they're not done separately from one another. We're also jointly maximizing the intermodality correlation as well. So that's why it's called parallel ICA. Just an example from some of the ADNI data looking at Alzheimer's and MCI. We found, in this case, one structural component that was linked in different ways to 4 different genetic networks, each of which had unique gene signatures and unique patterns of loading. So just again, a teaser for the kinds of things you can do last slide for this section, there's also been a lot of interest in looking at epigenomic data where, we have, for example, DNA methylation, which can change over time, essentially through the formation or removal of these methyl groups. And so looking at links between genomic, epigenomic, and brain imaging data, challenging problem. There's a lot to be do there. There's a lot to think about there. And so this is just an initial approach that we looked at with Dongdong Lin to bridge these together. And we found, for example, in the schizophrenia data set, some SNP methylation and structural MRI linked components, either two-way or three-way. And last one here, kind of it was already referred to by that question I mentioned earlier. But this is sort of a teaser data set where we looked at sort of a simple multi-layer perceptron with unimodal and multimodal branches, and we found that if we looked at structure by itself, you get you can see there's this bit of a separation between patients and controls there. SNPs results always look terrible. They're very small effects. They're hard to find. You need really large ends to find them. But if we combine imaging and genomics, we can actually improve our ability to separate and push back to get at, for example, relevance by looking at brain patterns and particular biological pathways that are implicated. Data sharing, population neuroscience will be led by Jeremy Bockholt, who's been doing this for many years. He was involved also in the initial FBurn project and has been working with us for years on coins. What are the repositories? What's missing? What do we need to innovate on in terms of reducing the barrier to data access and analysis? There's been a lot of movement in the field in this area. What are some of the ethical issues we have to think about? How can we best leverage large data repositories? Should we use them for pre-training? Should we use them to develop, how do we use them to develop predictive biomarkers, et cetera? There are some standards starting to emerge, in terms of data format. One that is being used a lot is called the brain imaging data structure. It's really just a way to organize your data so that it's a common framework. It can be validated, and you can pass it to someone else, and they can run an analysis without trying to figure out what you did. Analytics needs to be described in a more standardized way. One initial approach focused on linear modeling approaches is the neuroimaging data model, which has been adopted by some of the major software packages. So that's another example. Some of the data storage, data sharing tools that we've developed here at Trends include Coins, which is really a data capture. It's not really just data sharing, that's kind of the end piece, but it's meant to be a sort of a beginning to end study management tool, which handles all the privacy issues, data sharing, radiology reviews, et cetera. And can be connected to a scanner and automatically import and QC data. But it can also offer data sharing through a data sharing portal. Similar to that, we've more recently been building with Eric Ferner a tool called BrainForge, which is really containerized analytics pipeline portal. So essentially, you can take data and have it come in anonymized, and then you can run automated analyses on it. And so it integrates with Coins, but it can also be used in an unmanaged way. And you can run pre-processing, you can run group analysis, you can run ICA analysis and things like that. Another very complementary piece that we've been focused a lot on is decentralized analysis. So a lot of repositories have emerged over the last few years, but those only work well if you can put your data in that repository. And that is not the case for a lot of data. This might be due to regulatory concerns, rare disease issues, it can be legal issues, international boundary issues, you name it. But there's a lot of data that is not getting, and it's sometimes the most interesting data that is not getting shared, and that's not for lack of trying. We've tried for many years to share one of our schizophrenia data sets, and we have been unable to because the IRB will not let us do it. And so you can overcome that with this decentralized framework where the data stays where it is. You download a tool, you can map to that data. It never shares the data. It doesn't share any individual subject information, but you can have multiple sites do this, and then they can basically, someone can build a consortia, create a model, for example, a VBM analysis or a regression or a dynamic functional network connectivity analysis, create that model. It goes and it runs locally on the sites, and then group level features are iterated on, so it can do more advanced machine learning things, even support vector machine, and we're working towards, we've got neural networks, but even deep learning. So this is a really powerful framework that is very effective if you cannot share your data, or if you have lots of large repositories that don't coexist and you don't have the resources to put them in one place and you want to leverage them. Or a third example might be somebody's got a large repository, they've got QC on that data, it's very well evaluated and checked. And I want to compare how my data looks to that, and I want to actually use the data itself to do that comparison. And so that you can do that in a way that's sort of low bandwidth, right? It doesn't require you to create a large data set, a large set of resources. So I'm just going to skip over this, which is really just a sort of a vision to kind of say, well, we want to integrate centralized, decentralized, we want to ultimately develop dashboards and predictions and things like that that can help us advance our understanding of brain disorders, of healthy brain, of lots of things. I put this competition slide on there because this is just about, it's ended. We're validating solutions now, but the things that's happening over the past multiple years is more competitions are occurring. It really is useful because it gets lots of people looking at a common problem in different ways. Thanks, Matteo. So this was a nice competition, which was joint. It was the first UK Biobank competition that was a collaboration also with Trends, OHBM, and IEEE. And we actually got over 1,000 teams and 14,000 entries submitted for it. Deep learning. Are deep learning representations more predictive than standard machine learning methods? Do they record meaningful attributes of the brain? Are they robust? Can they be interpretable, etc.? And check out Sergey's chat there for checking out the deep learning discussion group. So, we started looking at this a few years ago, and our first question was, is this just yet another technique that's going to be, hyped up and not useful? And we found that it actually does look to be useful. If you look at things like prediction, for example, you see training and testing data both start to separate further as you go from depth, 0 to 1 to 2 to 3. But more interesting is if you look at sort of visualization, where each dot here is a subject, you see these very nice gradients of information that are sometimes learned without you telling the algorithm anything. This is some work from Anissa Bruhl that was looking at the question of, does deep learning outperform standard machine learning as across different sample sizes? This is some of the biobank data going from 100 to 10,000 subjects. We found looking at some very simple classification tasks, looking at age groups and looking at male versus female, and indeed, it using these two deep learning approaches did outperform consistently and significantly linear discriminant analysis, logistic regression, as support vector machine, various approaches. And here, as I was mentioning about features that were learned by the data, this is one I'll just kind of focus here, which is that Huntington's data. This is run by Sergey Pliss, and we separated control from patients, but post hoc we visualized the severity of the disorder, and we found that least severe was closest to the controls, and there was this gradient to most severe. And that was pretty striking because it actually learned that feature without us telling it anything about disease severity, and it sort of convinced me that there's a lot of potential value in these kinds of approaches. Jeff Mullins will be leading a group on cognitive neuroscience. This looks really interesting. Is there meaningful information in whether there's noise, what's previously been considered noise? I'd say yes, because we look at noise all the time. There was another recent paper by Lucina Uden commenting on this. What role should neuroimaging play in diagnosis and treatment of cognitive disorders? What are emotions? What is the function of sleep and dreams? Interesting questions. We know sleep plays a key role in functional connectivity. Just if you look at measures of EEG wakefulness, for example, delta, theta, alpha power, you can predict patterns based on those measures. You can also predict sleep stage from fMRI data and from EEG data. So that's just really a basic level showing that these variations, sleep state dramatically changes connectivity patterns, and that's just just touching the surface. A lot of interesting work coming out of Michael Chi's group as well in Singapore, looking at sleep and its impact. Enrico Premi will lead a group on neurodegenerative diseases. He's very interested in things like, moving towards a clinical diagnosis, centralized data-driven web-based framework. What would that look like? Single subject indices, longitudinal studies with complex modulators, environmental variables, genetic factors, and multi-center data harmonization approaches for resting fMRI as a starting point. Here's an example. There's been a lot of work looking at, Alzheimer's and MCI these days. This is just some work again from Anis showing that, it's relatively easy, to predict who has Alzheimer's from controls, but by then it's too late, to do much about it. We can interestingly predict, stable versus progressive MCI at an earlier phase, with an accuracy of an AUC of about 81%. Lots of people doing lots of interesting work in this area, and also looking at going down to young ages. And that's basically all I wanted to say. I'll just kind of put a plug here for we've tried to share some of our tools. We have a lot of the techniques I mentioned are shared in the GIF software, which includes, it's a MATLAB tool, but we also have standalone implementation, containerized implementation, and an IPy interface. The Fusion ICA toolbox and the other tools that I had mentioned, just some screenshots, from the dynamic connectivity toolbox. Here's the group at Trends, or at least a subset of them, and this is pre-COVID, I might mention. So we hope to get back together again in person soon. And with that, I will pass it back to Armin, I guess. You want to take over?
Thank you so much.
Yeah.
Thank you so much, Dr. Calhoun. So we are open for question if anyone has any question. I personally have one question. So I was wondering if there is a way that we can leverage those open sources and what Trend has for the NeuroMark and BrainForge, and do some kind of like a replication studies to see like if a specific approach works or not, if it has a diagnostic or not, a diagnostic power. Like I know about maybe we can. I remember we had, as you mentioned, about the Kaggle competition. So I would like to hear more about those if possible.
Well, others can answer maybe too, but I would say, yeah, that's definitely what we're trying to do. We've started again. The real challenge is coming up with clinically relevant classifications, right? And classifying schizophrenia from healthy controls isn't that useful clinically. But if you can find these categories like response to medication or potential, biomarker discovery, or we're working on one project with some psychiatrists to try to design, a dashboard that might help them, provide a tool for guidance. Again, there are a lot of things they look at in the clinic, and imaging could be a component of that. And it's underutilized, and it's, but, there's still a lot of work to do there. I think we need, and that's why we're doing NeuroMark, is because another advantage of that is it gives us a common framework that we can compare. I think it's really important to look across diagnostic boundaries, right? So we're not just looking at patient control. We want to get at specificity, We want to look at replicability, as you mentioned. We want to look at accuracy and things like that. So I don't know if others have thoughts, but those are just my quick thoughts.
Thank you.
Kind of the holy grail of
yeah. Is there any question regarding?
Looks like Elon had one.
Great.
Can you on?
I think.
I have
there you
one.
go.
I have one.
Yes, you're on.
Yes, I just have a question about how to get help. For example, I want to know how to use gift box, and say after reading the tutorial online and going through the weeks, still have some questions about how to use a gift box, a toolbox for specific purpose. So which person should I contact and reach out to for?
Oh yeah. So there's a couple ways you can do that. One is there's a listserv with Gift that you can email and you can search. And I'm not sure if that's visible or if you've seen that yet. And there's a bunch of questions people have asked already up there. So that's one. Another would be, I think that's exactly one of the things that we want to do with the BSI is provide a place for you to go where you can actually interact with real people and have discussions and say, well, how do I do this, right? Or what do I want to do there? And you can always just email too. So no worries at all. So yeah.
Yeah. And we welcome any question in any specific study group, either from the pre-processing or about toolbox or more sophisticated, or if anyone want to get others' opinion on their analysis or their result, we encourage all of those. And I hope everyone can feel we are all equal, we are all friends, we try to push the science forward.
Yeah.
I do have a follow-up question. I don't want to waste your time. I just want to find the person that I think would be most knowledgeable or most willing to answer my question. So, for example, if I have a question is in using Giftbox in analyzing my MCI patients' data and get a professional opinions of this. So, which person should I specifically look for? How do I? So my question is, how do I look for a person for a specific topic if there's a list? So for example, if Alzheimer's, I should look for this person. If schizophrenia, I should look for this person. Things like that.
Yeah. So I think that I would separate questions about how the tools work from questions about how do I want to design a study to ask something about a particular disorder, right? And those are both interesting. I would say there are, you could almost email anybody at Trends, right, about Gift. If you'd like, you can go to, again, the listserv is a nice way to go and get questions answered. And perhaps we'll have, if you're looking at, MCI or Alzheimer's, Enrico's got a study group that he's starting in BSI, and he's already been working with Gift and familiar with it. So he could certainly, I'll put you on the spot, Enrico, but yeah, you'll be the Gift expert there, and he knows how to, ask other people if needed. So again, I think what we're trying to do is build networks here of people so that we can, jointly help one another.
And I would like to add one more thing. If you are not sure which study group you can ask, everyone can ask any question in the general channel. So if you are not sure where you need to ask, where you should ask your question, when you ask in that general channel, people who are familiar are probably going to reach back, answer back. So we have that general channel for this purpose as well.
I have a question. This is Maria talking, if that's okay.
Yes.
So I also kind of wanted to, Armin, you mentioned pre-processing, and I feel like a lot of the study groups although they have really great topics, they're also really high level. And I think based on what you and Vince have said is the goal of BSI is to also make neuroimaging analyses accessible to people that don't have some of the resources that we have at Trends, where pre-processing is like not something that you necessarily focus on because everyone has a really good handle on it. I feel like having a pre-processing study group might be valuable as well, so that you can really serve the people that are kind of coming into the neuroimaging analysis, which it seems like is the intent of BSI as well. And I'm asking this also kind of to the group to just know if that is something that everyone would be interested in. If you you don't have to respond necessarily. I'm just throwing it out there because I've run into a lot of issues with pre-processing myself, doing a lot of analyses as a graduate student. And I think that it would be something that would be valuable to the kind of beginning level, I'm starting with neuroimaging kind of questions.
Great idea. I think it's the pre-processing has so many choices and so many pitfalls. And even if you've been doing it for years, you still fall into pitfalls, right? So there's definitely a great idea.
And also, I think just really quickly, I think maybe something that would be, I think it would be pretty easy for people to do too, is like if you've maybe starting some type of publications list of like, hey, I study Alzheimer's disease and I've used GIFT, or I study schizophrenia and I've used source-based morphometry, so that people who are looking at like where there's an overlap between methodology and disease might have a place and a resource to go to. I think that would be a really easy thing to just have a list of like, hey, these are publications that have done, like, here's a methodology and here's a disease, and like here's what they've done together, and here are the topics that are covered under these papers. I just think that that might be a really great jumping off point for people in BSI to kind of have as a really easy resource.
What about also like Armin, we could do maybe an initial Google form and have people enter topics or select topics or something, and have that available for people to search, something like that.
Sure, definitely we'll do that. We also are each study group also creating in the BSI website, they have like a page of resources for different paper and tutorial and others that we are gradually building. But I can also definitely create such a Google form and share with everyone. I think that would be very helpful.
Mm-hmm. Thanks, Maria. All right. Well, hopefully people think this is a good idea. We'll see how it goes. And we're new to this, so and we're not trying to be too proscriptive. We're just trying to kind of get things started, and we want some things will grow, some things may not grow, and we're open to changing, and we're open to updating. So please feel free to make suggestions. Any of your ideas are welcome. I'm sure Armin would love to receive those. So,
can I ask a question? This is Jessica. Is everybody who's attending aware of the Slack channels and signed up for those so that they're getting information? Because I know that's not a natural form of communication for a lot of people on the call. I don't know, Armin, if you want to post in the chat window maybe how to sign up for the Slack channels or something in case people are ready.
Sure, I will do it right now. Thank you so much for
Yeah, I was noticing that there's notifications of the first journal talks and things like that going around, and if you're not in the know, you're going to miss out. So it's.
Well, you saw Sergey's comment. He says it will become natural.
Resistance is futile.
Whether you want it or not, yeah.
But, no, I think the Slack channel is really important. There's actually is there a CABI Slack channel as well? See, I don't even know all the Slack channels.
Yeah. Vish, there is, right?
Yeah, I thought Jess was a part of it. I can see her here. So.
Well, like I said, it's not natural. I only use it on one computer, so it makes it a little bit tricky. But no, I think between Teams and Slack channels and things like that, people can get a little confused. So it's really helpful to keep reiterating how to access the BSI Slack channels and whatever's relevant.
Yeah, just Sergey's here. So there is a way to invite members to the channel just by link. If in the Slack channel we click on invite people, there is a link. People just can join by clicking on the link.
So if I may mention, add something. So everyone who is already joining to this study, to this talk, should have already registered via a Google form, via BSI website, and then we will add them in the Slack channel. And to receive talk information, including a link and user and password, you should be part of the BSI BSI team, or you should be a member of the BSI. And what we do is we send the email for the talks, and also we announce it on the Slack. So that's how we do it. And I will make sure that I will just going to share it right now in the WebEx in case someone hasn't received that. Thank you.
Excellent. Any more questions? Thanks, Harsh.
Well, I'm really excited about this, and I'm glad to see that other people are excited too.
Yeah, this is good. We'll keep this going, and hopefully this will be helpful, and it'll lead to some really cool things. Yeah, the BSI has tons of its own channels. Yes. Well, thank you, everyone. Armin, did you have any closing words you wanted to say before we signed off?
No, just want to mention again, reiterate that feel free to ask questions, chat in the BSI. We are all equal, and we hope we can have a nice conversation between different team and people.
Yeah, we are. This is flat, right? So we're not. It's not meant to be hierarchy, right? So we
exactly.
Just reach out.
Yeah.
Open.
And
we're all friends here.
Yeah, exactly. And I send the Google form in case you haven't received it in the WebEx channel as well. We also have a Twitter account that you can find information there. And I hope we can have a wonderful discussion on the BSI Workspace channel. And by the way, we have the first junior club for dynamic analysis next week on Friday. And also Sergey has been doing the junior club study group inside the trend, and we are probably having some of them in the BSI. And Jean and Jiayu will have another junior club for the genomic study group sometimes very soon. That's all. Thank you, everyone.
Simon.